I want to go back a moment and look at something. This is all about facilitated diffusion, just simple diffusion. Facilitated is when things move because they're being pushed by some other force. Now there's still, you don't need to burn energy to make this happen. There's still a gradient, and things still want to go from an area of high concentration to one of low concentration. What you see right there is a channel protein. The one over here was a carrier protein. And actually, this thing is like squeezing, carrying this object through. And still though, because you have so many out here pushing, it still requires no ATP. So those are well, that's one carrier protein from start midway to finish. This one is simply a channel, and it's like, this hollow, nice fast through it. Because of the protein, it can open and close, and it can shut the door. Ions are the ones that most often move through this. Glucose can move through it. Because glucose cannot go through the membrane. It's got to go through the little blue door, which is a protein. We still use proteins a whole lot. The things on both sides, of course, are the double layer of the cell membrane that objects cannot go through unless they're fat soluble. Then they go through it. But more things than not are not fat soluble, and that's why the cell membrane is a good barrier. Because if you're not fat soluble, you ain't going through that membrane. You might go through the blue door, but you're not going through the membrane. Now, oxygen can go through the membrane. Carbon dioxide can go through the membrane. Glucose cannot. Ions cannot. They have to be moved through the channel. And so I mentioned carrier and channel, and I want to show you the carrier too, so I did. These are the things that move via facilitated diffusion. Of course, glucose does, and small ions and amino acids. They move through that little blue channel protein. Notice that oxygen is not on there. Carbon dioxide is not on there because they go right through the cell membrane. These things can't go through the cell membrane. All right, now 15. Now, there are some specialized proteins that are just used to move water through the membrane, and those are called aquaporins. Cells use a lot of water. It's like many factories are sit on a river because many factories use a lot of water in their production process. Well, cells use a lot of water. And it's still facilitated diffusion because when you open the door, it just rushes in. You ain't got to make it come in. You just open the door and there it comes.
So, facilitated diffusion, the facilitated means there's something working to make it happen?
Yeah. But see, if we had a beehive out that door and the beehive is busted, the bees are swarming, right? Mm-hmm. They'd like to come in here if the door's closed. Open that door, here they come. It's because sheer numbers. But the door is what's keeping them out. So the door is what's going to facilitate it. If I open that door, they're going to come in. And I ain't going to make them come in, or come in on the board. That's facilitated diffusion. Now, if that door was off there all the time, and of course, you couldn't put a pickup truck to it, but larger, small things can go through. But these the little blue things you saw, they're not really blue, of course. But that's what's going to determine on the bigger particles what can come in and what cannot go in. And some things are still just too big to get inside there. The lysosomes, the mitochondria, there's no way they can pass through any kind of opening. Feel like an elephant comes to that door. He won't fit. So those are manufactured from within the cell. And they stay in the cell. That's where they come from. But to make those to make those particles, you're going to need fat to make them. I tell you what, if you ever look at a membrane that surrounds the mitochondria, it looks just like a cell membrane. I mean, 2 bilayers, and that's why we can teach the cell membrane as a standard membrane and then say, well, they're all like this. The nuclear membrane, same way. 2 phospholipid layers. I mean, without knowing which is which, you would not know whether this is a cell membrane you're looking at, or a nuclear membrane, or a membrane surrounding an organelle. They're all made the same way. 2 phospholipid bilayers. I reckon when that works so well, why modify it? It's used over and over again, and it's how it's used. But water, of course, does not like fat, and water does not pass through the membrane. So you have these little openings, open and close. But since they're geared just for water, aqua means what?
Water.
And pour means pour. So we have a name, I don't have a picture of one of those. But they, open and close, let water in, let water out. All right. Some processes that a cell uses to get things in and out must have energy. They can't just do it by themselves. What's it mean to move in a direction counter to the gradient? What's that mean? Yeah, so if you're swimming upstream, are you not going counter to the gradient? Water flows downhill and you swim uphill. Well, that would be, you got work to do that. Gradients will let things happen. Water flows downhill and you ain't got to make it flow downhill. If you're in that water floating, then you're along for the ride. But when you want to go back upstream where you came from, you got to paddle and kick and swim. That requires energy, and that's what we mean here. If you go counter to the gradient, if you go against the flow, I got to push you. I got to use energy. And on a cell, there's always more sodium outside the cell than inside the cell. That's how the cell functions. The sodium likes to go in and will be let in sometimes. But to make that cell go again, you got to pump the sodium back out. There's tons of it. That would be counter to the gradient. Now, cells have a number of ways to move things using energy. And I got some pictures on here showing you both some people's drawings and actual photographs through a scope. But you have passive transport, which does not require energy. You have active transport that does. Passive would be diffusion. Things just diffuse and I ain't got to make it. The bees I tell you out there, if I leave the door closed, they'll stay out. If I open it, they'll come in. And I ain't got to make them either. Well, that'd be facility, the door's helping me. But if that door had a big hole in it, then they won't come in anyway. That'd be just called diffusion. We'll try to fill up both areas.
So facilitated diffusion requires energy?
No.
It does nothing to open the door?
Moving the particle is not part of opening that door. It does take power to open that door. But when a door is open, you ain't got to do anything to bees. They're going to come by themselves. So even opening these doors, that requires energy. But that's not part of the process of shoving those bees in here. Now, if I got to actually go out there and make some smoke and drive them in, that would not be facilitated because I'm doing something to it. But the fact that the doors require energy to open and close. But that's not part of the process of moving the molecules through. If I got to shove those bees in here, I got to throw them in there, and that's different.
So is it right to say that the only one that requires energy is active transport?
That's exactly right.
Okay.
That's exactly what you want to do. That's what's called active force. It requires you to do something and make it happen. And like you swimming upstream, that's dead on active transport. If you're in a canoe floating or in a tube floating downstream, that's passive transport. You're doing nothing. The river is, but you're doing nothing. You're just along for the ride. If you want to turn and paddle back up it, then you got to do something to fight the gradient. Yeah, because you're going to counter. That gradient is downhill, of course, where it flows. Okay? Let's take a look at the first case. Active transport uses carrier proteins. And we're going to take that particle and put it where it don't want to go, where it doesn't want to go. They're always going to be working against the gradient. Always. Otherwise, you wouldn't need energy if you're working with the gradient. Is that many blank substance to be carried? Is that many blanks? Oh, it is? Okay. I didn't know if I had too many red words. The substance to be carried. And before I show you my picture, let me do a little talking about this one. There's always two carriers working here. One is going to actually move the substance, and the other one's going to produce ATP to power the first one. The first one can't make its own ATP and power it too. So the first substance, his whole job is to recognize what it is you want to move. The second carrier, his job is to make the power source required to make the other one do the job. They always work in pairs. And these protein carriers, it's not me, they're called pumps. They're called ion pumps. Because if you pump water uphill, if you pump water out of your well, it doesn't want to go, does it? You got to make it happen.
So the channel proteins are for passive diffusion and the carrier proteins are active?
That's right.
Say that again.
The channel proteins with a hole through them, when they open, in comes the stuff. That's just passive. But the carrier proteins, they're actually cranking it. But carrier can go both ways. And you carry it with the gradient or against the gradient. The channel cannot. I mean, if I open that door,
It's one way.
it's a one-way. The bees can't come in. Let them more here, let them out. That door can be a channel, be a carrier. I can carry things in with the gradient. I can carry things out against the gradient because a carrier does both ways. But a channel protein, it just opens up, and if there's a gradient, things move. If not a gradient, things don't move.
So the first you said recognize what you want to move, and then the second gives you the energy.
Yeah, the first one's going to be a carrier. The second one is going to produce the energy. Another carrier probably to produce the energy. But they're going to work hand in hand. Because I can grab the substance, but I can't move it until somebody helps me. It's too big. So. And they don't always look alike now. They don't always like that big old blue thing you saw. The picture I'll show you here shows you. If you look carefully, this right here, okay, this side is one of the carriers. Now, if you look carefully, this yellow jelly bean is ATP. Because when you plug it in there and you tear off the phosphorus, it goes back becoming A2P, right? And there's your phosphorus. These combine to give you a third phosphorus, but in that bond is where you find the energy. So, like this, it's open. So when ATP inserts into this receptor, then the energy is provided to move through, and then once it's through, a new ATP plugs in to do it again. All right? So this is one of the carrier proteins that's going to provide the energy to make this one send the product through. Now, in this case, this has the energy. This battery is live. And once I use it, it becomes this. Now the battery is gone. And once it gets past here, it comes on through. It's like me squeezing toothpaste out of a tube. Can't put it back, can you? I guess you could. I got put it here originally, so. But what I see here, this is the active site where this is going to plug in. And when it plugs in, it's got to then produce the power to drive the other carrier protein. And to do that, it tears off that third phosphorus. But now you no longer have ATP with 3 phosphoruses. You got ADP with 2 phosphoruses. And you got a third one that you can put back on there again and recharge that battery and do it again.
So that's what that other yellow P there?
This yellow here is the ATP, fully charged.
Right.
This used to be this. But when you tear off the phosphorus, then you get the energy you wanted.
Right.
And now to make another ATP, the mitochondria puts in energy, the third phosphate rehooks, and ADP plus phosphorus with energy becomes ATP. And the ATP contains the energy that you're going to need to make this thing happen. There's no power in this. There's power in this. And to get the power out of it, all you do is called phosphorylation. You tear off that phosphorus. And there's a bond right here, and out comes the energy. And this thing wiggles and changes shape and twists and squeezes, and this particle goes on through. All right?
So we're getting it. The way we get the more energy is from that particle that's coming back in.
The yellow particle has the energy,
Right.
and then that green particle and the yellow ball, to reassemble those and have more energy, the mitochondria must do that. The oxygen you breathe, the food you eat, goes in the mitochondria. And the mitochondria then oxidizes, it goes through respiration, and splits the glucose into 1,000 pieces. And as it splits the glucose, out comes energy from the bonds in the glucose. Well, the energy's got to be caught. It's going to get away. And it's caught in a bond right here. So when you attach this phosphorus to this thing, what you've done actually is you have trapped the energy in that third bond. But you have become ATP. ATP ships out to where you need energy, and to get it back, all you do is tear off that third phosphate, and out comes energy. And you can use it to make your hair grow or make things move. But now, you can't do any more until you reassemble those two and turn them back into ATP. And to reassemble them, you got to go one place, the mitochondria. Because they're the ones producing energy. You're warm right now because you can't catch all that you produce. Some energy still gets away, and you're warm because of it. But you catch a lot of it as ATP. And the ATP is what you got to have if you're going to power all these metabolic processes. That's this metabolic process going on here. You're moving something through. And when you take the ATP and you tear off the phosphorus, is that anabolic or catabolic? If you tear off the phosphorus,
Breaking it down.
so what would that be? Catabolic. Now, when you send those two back to the mitochondria, so it can put them back together again, and what he really wants is not, he wants the energy. And the way he does it is he makes ATP. But the goal is not to make ATP, it is to make a way to capture energy. It just happens to be a 3-phosphorus molecule doing that. You know the adenosine, that base that you can swap with, in the new DNA and RNA? ADP, ATP is adenosine, diphosphate. That's a nucleotide. That's a nucleotide. ATP, ADP, RNA, they're all nucleotides. And when we talk about you eat carbohydrates, you eat fat, you eat proteins, you eat nucleic acids. So you can make not only RNA, DNA, but these things right here, that ATP, ADP, those are nucleotides too. And a lot of folks, most folks know that RNA and DNA have nucleotides. But if you look at this thing with the structure, it looks like the nucleotides of DNA, because that is a nucleotide. That's the adenosine group tied to 3 phosphates. And the way the body works it, I can store energy in that bond between second and third phosphorus. So if I want to build energy, I need some ADP and some phosphorus. And I'll take the energy I want to save, and I'll use that energy to make that third phosphorus hook up. But then ADP has now become ATP. Now, that is my battery. Now I have it, I can use it when I want to. And then that battery goes to all kind of processes. And to get out the energy to drive the process, the body just rips off that third phosphate. Now comes energy. But to reestablish it and restore it, you must send both things back to mitochondria where they can re-release energy and take that third phosphate and take it back on. You need phosphorus in your diet because of that. Yes?
Is that an example of sodium potassium pump, or is it just?
That's not. Sodium potassium pump looks a lot like, have you seen these, the spinning, you go into a building and you go through like a spin, what's that thing called? It's like a turntable. And you
Turnstile.
walk through, and there's something that hold doors, and the whole door spins and shoves you in. Well, in that, you're spinning it, right? But in an ion pump, there's something spinning that door. And have you ever got ice cream and you, chew gum and you spin that thing and out comes the gum? Well, as you spin that lever, a little handle grabs the gum, right? And carries it down and falls out. That's like an ion pump. Something must turn the handle, that's you. And as the handle spins, it carries things with it from one area to another area. You can also use that to reload that. It'll send things up in there also. And that's the best example of ion pumps I've ever seen. Is the old-fashioned bubble gum dispenser, put in a penny, I'll probably down now.
Quarter.
You spin a quarter, and you spin that handle, and you'll see them move up there. And as it spins, it will just, it's like a revolving door. Take one down and drop it out. That's an ion pump scenario right there. Perfect. But in this case, that could send one uphill if you had like a line and you kept shoving it, they're going to start falling off, right? Ion pumps work against gravity. And the ice cream in the bubblegum dispenser, you're working with the gradient, right? There's a lot up here, I want one of them. And you just spin it to get one to come out. Well, if that was a big particle, like in a cell membrane, and you got to get it through there, that pump would spin like you just did and send one up into the cell. Now, if you had too many in the cell, it can spin and send one back out. But the spinning requires, it won't spin by itself. That requires ATP. Okay? Now, ions, like especially sodium and hydrogen ions, they're also used to provide energy. And when we discussed glycolysis, and we discussed the electron transport chain and how that works, you're going to see how ions can drive a process to actually make ATP. Because when these plants out here are doing photosynthesis, they're using sunlight and water and carbon dioxide, and what they're putting out is glucose to make more leaves and stuff. And they're also putting out oxygen. To them, oxygen is trash, but we need it. And to us, carbon dioxide is trash. They need it. So animals and plants, they actually go hand in hand. And the old-fashioned terrariums, where you could take and seal a jar. As long as you have some food, then things can live in there airtight, because the plants made the air, the oxygen. The animals made the... Well, this whole earth is a huge terrarium, if you think about it. Because everything I need, the plant makes. What he needs, I breathe out water. I go, there's water. So we're a perfect team. And that's what we're concerned about, cutting down the rainforest. If you start moving too many trees, what's going to happen to the environment? Who's going to make the oxygen I need? See the problem? And there are a lot of folks say, well, I need that ground to plant my corn. I got a family to feed. And the way it works where the rainforest is, the soil is not very fertile. So when they had a crop for a few years, they cleared more forest and plant. And over time, what's happened in the rainforest is going away. And we don't have much rainforest from the get-go, let alone cutting down. But that poor farmer, you got to feed his family. That's going to plant black-eyed peas and things like that. We tried to teach him how to fertilize the ground and leave the forest alone. Just add fertilizer. But that's an old trick, and that new dog's going to learn it. We've given them bulldozers and tractors. When they break down, they go back to the mules. Don't fix them. It's just a culture that we can't get past. Over here we have crop rotation and all. We use the same acreage over and over again. We do rotate crops because certain crops pull out certain things and put things back. So you grow black-eyed peas a few years in this acres, and then later on say, I want to put black-eyed peas over here. Let's put corn over here. It's called crop rotation. It works very well. You would think something that makes so much sense would not be hard to grasp. It is. All they do is cut down more forest. That gives you another acre to plant on, but it removes an acre of forest, and we're hollering, y'all please stop. But people, we can't make them do things in their own country. All we do is try to educate them, and sometimes it works and sometimes it doesn't. All right. So when we discuss those other high-level processes, you're going to see how these things actually work to drive the manufacture of ATP. Okay? Now, do you get what this is saying? This is cool. It's like saying to somebody, don't you have family in Florida? Yeah. Are you going to Florida this weekend to visit family? Yeah. Can I ride with you? Follow me? I mean, I can't go unless you go. Because since you're the H and you're going to go anyway, I want to hitch a ride with you. So when you get to where you're going, I can separate. That's what this couple means. There might be a gradient for hydrogen where there's 10,000 moving to an area of less concentration. And there might be a lot of me, but when I hook myself to hydrogen and he moves, then guess who moves with him? I do. And I might be moving actually against my gradient. But my car is locked onto hydrogen. So when hydrogen moves down its gradient, it might tote something with it that's moving up his. You follow me? It's kind of weird, but the best example is, I like to go to Florida, but I ain't got the money. I ain't got a car. If I find anybody does, you have notes by going to Florida, I'll buy gas if you'll take me. Yeah, it happens all the time. But in this case, the person who's going to Florida by himself is H+. He's going anyway. Whether I go with him or not. So I'm going, take me. I'll buy you lunch for you, and I'll do something. I won't be a total scrounge, but a lot of times they are. But when you hitch a ride and you couple yourself, well, if the ion is moving down this grade, he's going to go anyway, right? You just grab hold and go with him. That's what this is active transport, because you are the one that's attaching yourself to him, and then he's moving of his own power. Actually, I am moving up my gradient. Like if I'm here by myself in Georgia, and I got 10 temps living down there, according to diffusion, when I want to go there, where they want to come here.
They want to come here.
They want to come here. They want to spread out, right? So if I want to go and visit my family, I'm going to be going up my gradient. But if hydrogen has 50 people up here and one down there, he's going to go where?
Down there.
And I'm going to hook on and just, can I go with you? And I beg a ride. That's exactly what's happening here. The hydrogen is the vehicle that carries me somewhere. And I must use energy to attach myself to the hydrogen. And then he moves of his own accord. But without energy, I can't attach to it. That's the cool. When I first realized that, I go, that is sharp. It's tricking. It's like the person riding in Florida. If you're going anyway, I go with you. That's exactly what's happening here.
What are some of the things that we carry?
Some of the things that carry are your non-steroids. The calciums that can't go through by themselves, but hitch a ride. The potassium, hitch a ride. Not glucose. Glucose won't hitch a ride. It just waits outside the door. Door opens, it comes on through. That's facilitated diffusion. All right. So that's what it means to be coupled, in that you're getting by because you're hitching a ride on something else. You're moving in its natural direction, and you're being sent to where there's more of you, but it works. Something large oftentimes requires a membrane to change its shape. And actually, it's called invagination. Swallow the object. And that forms a vesicle. When the thing finally gets inside, and it is surrounded by a cell membrane, because you think about it, if I move up to something, and I just surround it, well, my cell membrane is touching it, right? And I surround, my membrane comes together, then this part, piece off, comes inside me. He is still surrounded by my cell membrane. So actually, the membrane that surrounds these vesicles looks like the cell membrane because it is. And it's going to come on into the cell, and the cell is going to utilize it. It's going to go cross cell. On the other side, it's going to fuse to the membrane, and then undevelop and spit out the trash. It's called pinocytosis. So I'm not really there yet. But the vesicle is formed. Have you ever seen an amoeba eat? Hi. It's like a blob. Crawls up and just surrounds what it's eating. Next thing you know, it's inside the amoeba. But you still remember now, the part of the amoeba that touched that food particle is the cell membrane. So when it pinches off, the membrane of the vacuole is still that cell membrane that was there before. And that's why we've come to realize, we now know why all these vesicle membranes look like the cell membrane. It is. It's how it took it in. You're going to see a picture in a minute showing you better how it works. Now, these changes in membranes, they never stop. They're formed, reformed, unformed, deformed. They never stop. Your cell is a dynamic, moving thing. It's not like a shoebox that never changes. This cell changes shape all the time. And there are pieces of cell membrane that are being laid down and being done away with. One more thing in pictures, I saw some pictures on eBay showing, not eBay, YouTube showing that. Now, you can also spit something out. That means be exported. The vesicle comes up on the backside of the membrane, fuses to it, and then opens up, and then the trash that you've developed in the vesicle is released to the environment. In your case, it then goes into the lymph and goes to your lymph nodes where it's destroyed by white blood cells. I need to make red pancreas. I think that's going to be red, I guess.
Yeah.
I just happened to see it wasn't red. And exo means the exit. Cytosis means cell. So the process that exits the cell is called exocytosis. What do you think endocytosis is?
Inside.
Yeah, getting in. I mean, the words are easy to figure out. when that amoeba crawls up and surrounds and takes in, that'd be endocytosis. And when it's done with that food particle, it's got to get rid of it. So it kind of spits it out the other side. That'd be exocytosis. Now here's a picture. On this side is a person's drawing, but on the other side is the actual photograph. They caught it just in time. And it's like a balloon's blowing up. Actually, it is releasing to the outside. And notice, if you look right here, you see your double layers? They're right there too. Because over here, when I formed this, I used 7 million to form it. So now, when they fuse and come together and open up, then this is the trash that you're trying to get rid of. And the real picture, it looks remarkably similar to this. And that when it opens, the trash is spit out into the environment. But that's an example of exocytosis. Now somewhere over here, this thing was formed, taken into the cell, and if you look carefully, there's orange. See 2 orange layers? They're right here. This is a cell membrane that formed when we first made this vesicle way over here when it first went in. It's still cell membrane, but that's called vesicular membrane there. So when I show you a membrane, it always looks like, you know, the phospholipids and tail sticking down. It's always that way. It's always that way. And it's going to grow on up and become continuous, and before long you'll find no trace of this pit. It's going to be smooth again. So these are your stages. But like I said, right here, they're going to merge and eventually open up and let the debris exit the cell. Endocytosis, there are several kinds of ways to take something in. There are more ways to take it in than to get rid of it. I already mentioned to you pinocytosis. There's also receptor mediated, and I'll show you that one how that works. And there's one called phagocytosis that white blood cells do. All of them, though, get something into a cell. All 3 of those take something in, because that is what endocytosis is all about, taking things in. And like there are 3 ways, and you're going to see 3 different looks. All right, can I click it yet? Y'all ready? Okay, here's your first picture. This is, time is going this way. The cell just invaginates. The cell membrane does. Finally, these 2 areas touch. Finally, they become continuous. And now this is now inside. And if you look carefully, this membrane is the same membrane that surrounds these particles. It's a cell membrane. And any membrane we look at looks like with the phospholipids with the hydrophobic tails toward the inside. Because this actually is that. And it goes across. Now, certain things they can digest these, use them, but this is a kind of endocytosis. Of the 3 kinds I want to show you, this one really is the one called pinocytosis. Because with pinocytosis, you take a lot of water in, there's some things dissolved in the water.
This one.
Phagocytosis, you take a big old chunk in. like a chunk of bacteria. You got some water, but you got mainly a chunk of something. This one is like taking in Kool-Aid, where you have water and you got things dissolved in it. Here they're showing you dissolved particles real big. But this whole thing is still, I mean, this is inside the cell here, and this is extracellular. And all we're doing is just taking that cell membrane, we're making a pocket. We're making these two ends come together, and they fuse and become continuous. And now this thing is now called a vesicle. And it all happened because of a thing called endocytosis.
So this particular one you said is more.
That particular
Phagocytosis.
one would actually be the one called pinocytosis. That's not the receptor mediated, and that's not the phago. You'll see a difference in a minute. You'll be able to see them and tell. All right.
The next one.
Now I'm going to talk to you, and like I just showed you this, and I want to show you another slide of it too. But invaginates means to grow inward of its own accord. Substances then fill the cavity, like you just saw a minute ago. There's often fluids flowing in, carrying dissolved molecules in the fluid.
You fill or is this fall?
Huh?
You want fill or fall?
Fall. What did I say? Fill?
Fill.
Fall. Enter. Get sucked in. I don't know what to say. They're inside, as you saw earlier. I don't think they fall because there's no gravity. You can't fall up. I think they're actually, because they're sitting there, when you surround them, they're there. There's no, I'm just going to surround you. And I can take a fluid in that way, or I can take, like the flu shot in that way. You just actually surround all stuff and take it in. And then the flu shot actually is designed to destroy the virus. That's how it works. The virus takes it in. That interferes with the virus reproduction. And also, in the flu shot, there are baby viruses that either are weakened or they're dead. And your body sees them and thinks, oh my, here's the flu, and you start fighting, and they're dead. So when the real bug comes along, you're so strong, it can't hurt you. That's what the vaccination is for. Y'all know vacca comes from the cow, right? The first ever vaccination was for cowpox. It was designed to defeat smallpox. Dr. Edward Jenner, an old country doctor like Doc Adams on Gunsmoke, he began to realize that in England they had milkmaids who milked the cows. That was their job. And they would get sores on the hands sometimes. That was caused by a virus, and they call it cowpox. He then began to realize that all these ladies that had cowpox in a village full of smallpox, they never got sick. Now, no one thought about that but him. He goes, I wonder if having had cowpox might make them immune to smallpox. And he began to think there might be something that they got sick from cowpox, made all these antibodies that kill the cowpox, but it also works on smallpox too. He was right. He was so sure of himself, he went down to the local hospital, and first off, he gave himself cowpox.
First, he tried it on an 8-year-old boy.
Oh yeah, he wasn't real brave.
Yeah.
But when he wanted to put his money where his mouth was, he actually did it to himself, and he actually exposed himself to smallpox and never got it. And because it all came from a cow, vacca in Latin means cow. So a vaccination is the fact that the first ever vaccination comes from the milkmaids who didn't get smallpox because they already had cowpox. So he gave you cowpox on purpose, so you would not get the other one and die. And he was the first one to ever try that. And Edward Jenner, but that's why it's called a vaccination. It means of a cow. A lot of folks think, that's not very prestigious, of a cow. But that's where it all came from. Had not been for the milkmaids and their cowpox, and then they're not getting smallpox, we might not have vaccinations this very day. But you're right, he didn't try it first, did he?
Yeah.
All right, so this is another picture And I want to show you the real thing. It's showing you what's happening. And again, here's your cell membrane that's just wrapping around. This is still cell membrane. It's just now formed the membrane of the vesicle. Now here's the real deal. This is a cell. And these are the invaginations. And there's one that's inside. That one's like it's almost inside. And that's the other side releasing. So this would be going in, and that would be going out.
What is that measurement?
This is a pseudopod of an amoeba. It's an arm of an amoeba.
She said the measurement.
Yeah, I'm trying to figure out how small is that.
If you take a millimeter, and you know how that's small, right? All right. 1,000 divisions of those is one of those. Now you take something as thick as a dime, and you slice it 1,000 times. How thick are those slices? Not very. Well, right there, that's half of one of those slices. Can you imagine that? How do you get a knife that skinny to cut something that thin? My knife would butcher it all up. But that's what that means, though. That's the nanometer.
And that's an amoeba?
That's the amoeba pseudopod. That's not the whole amoeba. That's just an arm of it. And the whole amoeba is doing this, not just its arms. Okay? Now, this one is the one that's the new kid on the block. We just figured out how they do the receptor mediated thing. And then you catch up with it. Now I'll explain what's going on here. The cell actually lures a substance to it. Strangest thing. Now you're going to soon see this vesicle doesn't look quite like the ones I showed you earlier. There's one more thing going on inside it. It's a receptor. The name is great because you're going to soon see what the receptor is doing. It's a tricky thing. It's luring something in. And the picture will probably make more sense than all the writing does. But this is still active transport because the cell is working hard to make this happen. It doesn't happen by itself. Well, neither did pinocytosis. It didn't happen by itself. The cell's making that happen. And this is an example of the cells making this happen. I got through the red parts. I'm going to click it and see the picture because the picture is going to make it more sense to you. Here's the picture. Now, let me explain what's going on here. You see these little Y's right here? And the Y's right there? You reckon they're going to be attaching to the red marbles or the triangular things?
Triangle.
The shape matches, right? But those are the receptors luring, attracting those triangular particles. Now, they're going to land right here. And when enough have landed, see, there's some more there. There's some more there. So there's a round one here. Because they're going to come in anyway. But these are designed to catch the round ones. They go in just by happen to be there. But these triangular ones, they're being attracted on purpose. And when this is the protein coat that forms around the outside of it that supports these receptors. And when you pinch off, you still got this protein coat. That's a coated vesicle. It's like a little fur ball. Inside there, you'll see there's your receptor, and there's the particle. A few green ones are in there because they got just stuck in by the wash. But if you look carefully, you'll see them here in the real picture. There are. Now I can't see them this pretty, but there's the coat. Right there's the cell membrane. That's right here. And the protein coat is out there. And that's a coated pit. And that's your, if you see that furry looking thing, that's your receptor mediated process of endocytosis. And the whole thing is actually attracting what he wants. He's not waiting for chance for it to blunder by. He's saying, I'm going to catch this fish. And they're actually attracting him. Now, if you had a square looking object on there, he would not fit that receptor. Because the receptors are special for what they're trying to get. But they're drawing them in, and then when you have enough of them attached, it pinches off inside, and you have a vesicle. It's a vesicle, but sure does not look like that vesicle, does it? On this one here, it's just luck of the draw. What comes in and what I get. On this one, I'm attracting particular ones I want to have. That's your receptor mediated. I will still get a few oddballs like the red marbles, but I want those triangular things. That's the whole reason I want the triangular things. And that's what the cell does that for. And that's your receptor mediated. Because these little Y's, it's all about them. They're the attractor, they're the bait that this thing lands on. And when you have enough of them inside here, he'll pinch off and go into the cell. And then those will be used by the cell for making new structures or for metabolism, things like that. But that still requires energy. That just does not happen by itself.
And so the other one, it always just gets whatever comes in.
Yeah, whatever comes along. You see the triangles there?
Mm-hmm.
There's some in there. Why? Were they drawn or happen to be in the wrong place at the wrong time? Wrong place, wrong time. They just flow in. But now, if you actually want those triangles and you want them and you need them, then you move to dispersion and you'll attract those triangles in on purpose. Now, if you look at them, which one has a better chance of getting triangles? Plain old pinocytosis or receptor mediated? Well, obviously, you're actually drawing them in. I mean, that's not, you want them that time. You're going to get some this way, but it's just by look at the draw. I see one in there. I see one in there. I see three right there in that one. That's not waiting for chance. He's saying, come here. You're going with me. All right? Now, phagocytosis is the third type of endocytosis. This is something big like a chunk. When I did this, I thought of Haley's chunk of cheese she always gives us. That's a chunk. And this is like an amoeba. See how the word amoeba is spelled? Italicized, right? That's a scientific word. You must always italicize scientific names of critters. If I say I'm Homo sapiens, I must italicize those two words. Now, and I'm right, I can't italicize there, can I? So then what must you do? Underline it. The rule is, if you ever write a scientific name like amoeba, you underline it. Now, if you have a typewriter or word processor, then you can slant it italicized. If you have a regular typewriter, then you underline it. I ain't used a regular typewriter since I don't know when.
It's critters and what else?
Scientific names. Any scientific name. The few I know of, like these pine trees out here, those are your short-leaf pines. They're called Pinus tata. You have Pinus tata, Pinus alba, Pinus lustrus, Pinus globulum. That always has Pinus, right? That's like saying Tef Bobby, Tef 80, Tef Donna. My last name is Tef. Well, that's my genus. Pinus this, Pinus that, Pinus this. That's the first word you say. I mean, there's those Homo sapiens, What's another Homo y'all know of? Homo erectus? Homo is a genus. I'm not the one called erectus. My forehead ain't sloping. I got a fairly large brain case. But when you name these trees out here, maple trees are always acer something, because in Latin the word acer means maple. You had red maple, and rubrum is red, so you have acer rubrum. The silver maple is acer, I forgot the last name now. But the first name always means maple, and the second name is what kind of tree it was. Red maple or silver maple or blaze maple. It's always acer this, acer this, acer this. So when you're writing these things out here, when I wrote down like this tree is an acer, I got to underline acer.
So you only have to underline the Latin or Greek word.
Well, not scientific name. It's about scientific name.
Okay.
They're always written either italicized or underlined. So if I say we belong to the genus Homo sapiens, you say Homo sapiens. If you're typing it, you just slant both words. And the second word, you never capitalize. It'd be like Tef with a big T, Bobby with a small b. My brother's Eddie, it'd be Tef, big T, Eddie with small e. My sister Donna, it'd be Tef, Donna. And in Latin, you always put the noun in the front of the adjective. You know it, right? Well, acer rubrum means maple red. Well, I'd be saying red maple. And I think Spanish does that too, though. They swap the adjective and the noun. Instead of saying red car, they say car red. So that's English. By saying red car, we're the oddball. To me, it makes more sense to say that's a red car than saying that's a car red. But in Spanish, I guess French too, you would say that's a car red if you spoke the language. Well, there's a picture of something pretty large being drawn into a cell. You still make a vesicle, and you still actually the cell membrane that makes the wall of the vesicle. So that's a pretty large chunk of food right there that's been taken into the cell. This is an amoeba over here. This amoeba is eating this bacterium. There's the bacterium, and there's the amoeba surrounding it, and right here is where the stomach is dissolving this bad boy. A pseudopod means like a false arm. And it's hard to see it, but this is a rod-shaped amoeba, bacterium, and this protist, called the amoeba, is just like a blob, it's just surrounding it. And before long, the bacterium will be inside that amoeba, and then that bad boy is going to digest it. Your white blood cells work the same way. A white blood cell comes up to a bacterium, and the white blood cells and your white blood cells crawl. They crawl through your body. Can't feel them though, but they're moving around. When you get pus and a pimple, they crawl there because that's where the fight is. And you match it, out comes, that's white blood cells you're matching out. And they're white. You can actually mash too hard and drive the infection deeper in. And that's when you wish you never touched it because it gets worse on you. But sometimes you feel the pop and it'll be out and you'll be done with it. But in this case, this is the white blood cell. That is something that's got to be gotten rid of by the body. The white blood cell just surrounds it, takes it in, and then this will be dissolved by enzymes inside the white blood cell. And when it's done, there's nothing left. When white blood cells kill bacteria, upstairs in the lab, even I still got a mess to clean up, right? After I clean up, I still got a mess in the sink. So I clean that up last. Well, the white blood cell, it takes in there's some white blood cells that make the environment hypotonic to the bacteria. If you're hypotonic, you swell or shrink. Hypo. You swell. Now, when that sucker swells, he's going to finally explode, right? And you got bacteria pieces all over the place. But one of your white blood cells blew the sucker up. Who's going to clean your mess up now? There's bacteria guts all over the place. Other white blood cells come in then, and they clean the mess up. This is one cleaning up the mess of a bacterium that's been done in by another white blood cell. White blood cells, one white blood cell, I'm going to kill you. I'm going to make a mess. And when I get done, brother, you come in and clean the mess up. They do that all the time. This one here is cleaning the mess up. That's not a bacterium right there. That's a bacteria part that's got to be gotten rid of. Now, they won't get rid of it totally. And when you're done, the wound is clear because your white blood cells did it. But white blood cells can actually leave your capillary and crawl like an amoeba to where the injury is because your cells release chemicals like a beacon and that attracts the white blood cell to that area, and that's where the fight's going on. So as some white blood cells are killing bacteria and leaving the mess behind, other white blood cells coming in and clean the mess up. When you get done, you have no sign of or anything with it all. So you got 5 kinds of white blood cells, 5 different kinds of them. And if you have an allergy, it's your white blood cells doing it to you. You do it to yourself. If you're allergic to cat dander and hair takes you out, you do that to yourself. The white blood cell actually, in doing its job, makes you sick of the dogs. If your white blood cells weren't so sensitive, you wouldn't have the allergy. And some of your medications to fight allergies actually turn the white blood cell off that gives you the allergy.
So this is done by chemical reaction.
Exactly it is. Exactly it is. All right? Water. Can you overwater a plant?
Yes.
Yeah, you'll kill the sucker too. When they flood an area and back up the water from building a dam, many plants are going to die because they can't stand that much water. What's it mean to be differentially permeable?
Let some stuff in, doesn't let some in.
Some things can come in and some things cannot. Like a spring door. You got that in. All right. Unfortunately, my screen can't change its size, can it? But your cells can. Your cells can change their size to become even more permeable or less permeable. My screen door is set and is welded that way, and it can't change unless you poke a hole in it. And I know some folks who did a dumb thing. There's an air filter in your car, right? And the air comes into your motor, and then the air is combined with the gasoline. That explodes by your spark. It gets real hot, spark plug exploded. That makes the power turn the pistons. They turn the shaft, and you go, right? Now, the more air you put in there, the more power you have. You seen the big old scoops on front of cars to suck air in? Well, my Harley has one. My radiator, I have a scoop now to get more power. Because if you blow on a fire, it'll blaze up more one. Well, that's the idea here. If you can take and you can change that permeability, you can even speed up metabolism faster. But it all still boils down to those gradients. You know, the gradient, like downhill, uphill. Again, downhill gradient, I can coast, can't I? Uphill gradient, I got to apply the gasoline and put power. I know when I was real young, before I got married, I was not always as well off as I am now. I was strapped for money. And I would drive somewhere and I'd coast down the hills. In my mind thinking I was saving gasoline. Of course, back then, gas couple whopping 50 cents a gallon. If we had $1, I was going to stop buying it. That's shit. Who am I kidding? But it's all about the gradient still. And the gradient makes the difference between which way it's going to go. All right, let's see what we got here. There are 3 terms that describe the ratio of water to the solvents. We're dealing with osmosis. And these next 3 slides, when I was where you said them, they drove me crazy. I never could remember which one went. And one day I invented a method that is strictly Bobby Tail. I've shared with other teachers, they use it, and it's so simple you think, well, that's no big deal. It works. So I want to show you some, and we're discussing hypotonicity. Let's say hypo, iso. All right, so let's go ahead and just dive into it. The first thing is the word hypotonic. Now, go ahead and get the blank. I got to tell you something that you better not ever forget, because I used to forget it, and that's what drove me nuts. I think I'm missing a period. I think I'm missing right here. I see my capital letter. Yeah, I like that better. Now, when I was in school with you all, I tried to memorize it, and I just couldn't get it down. First thing I had to realize, when I began teaching, I had to know it, so I've learned it now. The word hypotonic, my first question is this. Is that referring to the solution or to the cell? That's the thing you gotta figure out first. Hypertonic.
Tonic is like a solution.
For the solid.
It's a solution.
Yeah.
What do you think?
So we refer to hypotonic solutions, hypertonic solutions, and I didn't realize that kicker. It is not about the cell. It's about the solution. Now, if you keep that straight, you better than I was in school. I never picked on that one. Now, the prefix hypo, what's it mean?
Lower. Low.
Huh?
Hypo.
Little bit, not much, less, right? So it means not much. Now, my next thing, you see that word T-O-N, ton? There's not much stuff in a hypotonic solution. There's not much tons of stuff. So, if there's not much stuff in a hypotonic solution, there must be more in the cell, right?
Yeah.
If I take this cell and drop it whoosh, into a glass of water, and I said that solution is hypotonic, I'm comparing it to the cell in a way. And I'm saying that there's less stuff in that solution, therefore there's more stuff in the cell. Now, the question now is, why does that cell swell up? It will swell up and burst. If you put that cell in a hypotonic solution, now, there's more stuff in the cell, less stuff in the solution. Now, I got a picture coming up. Yeah, let's do that. Okay? Actually, I don't like that. Oh, good. Okay, let's, I want to do all three at one time, so I'm going to stop here. I want to do one and let time pass. So, I want to come back on next Monday and pretend I'm starting here. And we'll move again. So, I'm glad to come back. I'm about to get into deep water.